All right, welcome back, fellow scientists. So today we are still on learning objective number 1, looking at the history of DNA. Last video we looked at Griffith and how he identified what he called his transforming factor, right? Now, a little bit of big overarching biology knowledge. So the experiments that we talk about in biology and AP biology basically fall into 3 categories. Category number 1 is a category that I call, that's interesting. That would be Griffith's experiment, right? He wasn't studying DNA, he wasn't looking for a transforming factor, he was studying pneumonia. For some reason, he mixed 2 things that shouldn't cause the mouse to die, and the mouse died. That's interesting. There must be a transforming factor, right? Today, we're going to look at the second type of experiment, which I call a labeling experiment, right? We can label with glowing proteins, we can label with dyes, we can label with radioactive material, which is what Hershey and Chase are going to do in this video. They're going to label something with radioactive material, and they're going to see where the radioactive material goes. So that's a labeling experiment. Third type of experiment we don't really get into in regular biology. This is more of an AP biology experiment, but it's called a knockout experiment, right? If you want to see what something does in a cell, you knock it out, and then you see how the cell behaves. So those are the 3 basic types of experiments. Now, before we get to Hershey and Chase, we have to cover a lot of background, honestly. So if you'll remember from chapter 2, we looked at the different types of macromolecules, lipids, carbs, proteins, and nucleic acids. So scientists knew that all cells had these, and so one of these 4 had to be the transforming factor. Okay, now they could immediately rule out lipids and carbohydrates from their list of potential candidates because they don't have enough variety and they're not found in the nucleus, right? They could look at the cell underneath the microscope, just like we did in our onion root tip mitosis lab, and they could see that the nucleus was evenly divided during cell division and each daughter cell got a nucleus. So it must be really, really, really important. And they could tell that inside the nucleus there were proteins and nucleic acids. Now remember our diagram of the chromosome, right? This is a nucleic acid and it's wrapped around histone proteins. And so it's really difficult to separate these 2 to figure out, okay, what's more important? Is it the nucleic acid or is it the protein? Okay, now put yourself back in the mindset of someone in the early to mid 1900s, right? They knew about chromosomes, they knew about the different nucleic acids and proteins, and they knew that proteins basically had 20 letters in their alphabet. There were 20 monomers, 20 different types of amino acids that make up proteins. Okay, they knew that there were only 5 types of nucleotides, which are the monomers that make up nucleic acids. So they asked themselves this question, okay, if I wanted to control a very complex thing like the cell, would I use an alphabet that has 20 letters or would I use an alphabet that has 5 letters? Well, obviously, they chose the alphabet with 20 letters, or they thought that it should be the alphabet with 20 letters because you can make more words with 20 letters than you can with 5 letters, and you can encode more information and more directions and instructions with 20 letters than you can with 5 letters. So most people thought that the transforming factor was proteins, not nucleic acids, right? Hershey and Chase, I don't know what their hypothesis was, but they probably set out to prove that the transforming factor was proteins and not nucleic acids. Again, not sure about that. I'm just hypothesizing. Maybe you can look it up and comment below and figure out for sure what their hypothesis was. So that's a little bit of background. Now, a little bit more background. Remember from chapter 1, when we were studying the characteristics of life, we studied viruses, and we found out that viruses actually have fewer characteristics of life than fire, but some people consider viruses to be living, and some people don't, right? And one of the reasons that some people don't consider them to be living is that they're so simple. They don't have all 4 classes of macromolecules. They only have 2. They only have the DNA, and then they have the proteins, right? And so that's very fortuitous for our purposes, because those are the macromolecules that we're trying to distinguish between, right? And so this is how a virus works, is that it will attach to a cell and it will inject something. They didn't know before Hershey and Chase. They didn't know what it would inject, right? But it injects something, and that something takes over the cell. The cell doesn't behave normally. Instead of doing what cells normally do, now the cells make more viruses. So the virus transforms somehow the cell. It changes the cell into a little virus factory, and then it makes more baby viruses, and then the cell explodes, right? The biological word for explode is lyse. And so the cell lyses, and then we get more baby viruses that go out and infect all the other cells. Okay, so that's a little background on viruses. Now, finally, we are to the Hershey Chase experiment. So this was around 1944, and they used this idea of viruses only having bacteria and proteins, and they also used this idea of isotopes. Now, back in chapter 2, remember an isotope is an atom that has gained or lost a neutron. Okay, and they took advantage of the fact that DNA has phosphorus, which normally has an atomic mass of 31, and proteins have sulfur, which normally has an atomic mass of 32. Now, DNA and proteins, they have a lot in common, right? They all have carbon, they all have nitrogen, they all have hydrogen, they all have oxygen, but proteins don't have phosphorus and DNA doesn't have sulfur. So that's the difference that they exploited, that they took advantage of. Okay. They also took advantage of the fact that there is a naturally occurring radioactive isotope of phosphorus that has an atomic weight of 32. And there is a naturally occurring radioactive isotope of sulfur, which has an atomic mass of 35. Okay. So what they did is they grew viruses and they only provided them with this radioactive type of phosphate. So in essence, they were labeling the DNA with the radioactive phosphate. Right? And then they took a separate batch of viruses and they grew those viruses that only had access to the radioactive sulfur. And so essentially they were labeling the proteins with this radioactive sulfur. Okay, so once they had these two batches of viruses, then they took the viruses where they labeled the DNA and they mixed them with bacteria. Right, and they, let them sit for like 20 minutes and let them do their thing, let them inject whatever they were going to inject, right? Then they put the whole thing in a blender and they pressed frappe, which, if you'll remember from our chicken liver lab, is my favorite word regarding blenders. So they pressed frappe, and what that did is that separated whatever was left on the outside of the bacteria from the bacteria. And at this point, the virus had already injected whatever it was going to inject into the bacteria. So then, after they pressed frappe, then, you're welcome, Will, then they spun it around in a very high-speed merry-go-round called a centrifuge. And what that does, it separates things based on density. Okay, so the cells are heavier than the viruses, and so the bacterial cells are gonna be kind of squished to the bottom, right? And the viruses, whatever was left outside of the cells and was separated by the blender, they're going to be up here in the liquid. Okay, and then of course they did the same thing with the viruses that they labeled the proteins on, right? They incubated them with the cells, frappe, and then the high-speed merry-go-round, and then they analyzed, then they got their data, right? So their question was, where is the radioactive molecule? Okay, if they found radioactive phosphorus in the bacteria, that meant that the virus injected DNA. If they found radioactive sulfur in the bacteria, that meant that the virus injected proteins. Okay, so you can see from this chart the results of their experiment. The virus injected DNA because the DNA was found in the bacteria. The virus did not inject protein because the protein was not found in the bacteria. In fact, it was found up in the liquid, what is known as the supernatant. Right? Supernatant, sorry. So one last diagram to kind of help you visualize this very elegant, very classic experiment. So they took one set of viruses, labeled the DNA. Viruses landed on the bacteria, injected the DNA, separated the viruses with the blender, and then they saw that the radioactive material, the radioactive phosphorus, was in the cell. Okay? Separate batch of viruses, they labeled the proteins. Viruses attached, injected the DNA, separated the virus from the bacteria, and they did not find the radioactivity in the cells because the radioactivity was in the proteins, and the proteins are not the transforming factor. Proteins are not injected into the bacterial cell. So this has been Mr. Leo. Today we have looked at the classic experiment called the Hershey Chase experiment, where DNA was without a doubt identified as the transforming factor. Thanks so much for watching, and I will see you on the flip side.